Once again, welcome everyone. I have here with me Dr. Darleen Sandoval. She is an assistant professor of surgery at the University of Michigan School of Medicine. She received her PhD in exercise science at Arizona State University and a postdoc at Vanderbilt. Her research really focuses on two general themes. The first one focuses on the role of GLP-1 in the regulation of energy and glucose homeostasis, and her second focus is really on the adaptations of the gut-brain axis with bariatric surgery and how that would contribute to the profound weight loss and improvements in glucose and lipid homeostasis. The agenda for this morning is essentially going to be, we're going to start off with her talk and some of the work that she's been doing in her lab. And then I'm going to segue into talking about ALPCO's new chemiluminescence active GLP-1 ELISA. And then at the end, we're going to open up the session for some Q&A.
Okay. So I would first like to thank everybody for attending this afternoon, or I guess it's morning on the West Coast, and for your interest in our work, and thanks to ALPCO for sponsoring this opportunity. So just to start out, I'm going to give you a little bit of background about the kind of work that my laboratory does. So we do preclinical work in mouse and rats, and the idea, I think, of any physiologist is that the work that you do in the lab translates in some fashion to the clinic, and whether this be spurring different clinical research studies or whether it actually impacts therapeutics is something that we're keeping an eye on in terms of our over-encompassing goal of the lab. But on the flip side, we're also interested in looking at clinical phenomenon and taking it back to the laboratory to see if we can understand either mechanisms of action for drugs or surgery in this case, or to see if it can inform more about what we understand about the underlying physiology. And so again, my laboratory is specifically focused on GLP-1 physiology, or glucagon-like peptide 1, which I'm assuming you know a thing or two about given your interest in the webinar. And interestingly, GLP-1 has been used very effectively therapeutically to treat type 2 diabetes and now also as an obesity therapeutic. And then the other thing that's interesting is that GLP-1 goes up tenfold with bariatric surgery. And so what we can do is we use a lot of our preclinical models to try to understand the exact mechanism of action of these therapeutics. And what I'm going to talk to you about today is what is the role of GLP-1 as a mechanism for bariatric surgery. So let's just start with a little bit of background, and that would be the conventional idea or concept surrounding GLP-1 physiology, at least when it's secreted from the gut. And so the idea that is that it's prandial hormone that is secreted into the circulation by intestinal L cells. So these are specialized enteroendocrine cells that are located within the distal gut. And once GLP-1 enters the circulation, it binds to receptors in the pancreas, and that causes an increase in insulin and a decrease in glucagon. And this change in hormonal milieu has a very potent effect on the liver, restraining postprandial glucose homeostasis. So again, this is isolating the impact of GLP-1 on glucose homeostasis, and of course there are many other effects. The problem with this model of GLP-1 physiology is that GLP-1 actually circulates in really low levels, and that is because it's rapidly intravascularly metabolized by an enzyme called DPP-4. There are also few, if any, there are few, if any, GLP-1 receptors located on alpha cells. And lastly, receptor signaling is apparent even when plasma levels are low. So for example, if you take pancreatic cells out of the body and put them in a petri dish and you add a GLP-1 receptor antagonist on there, you can actually inhibit cyclic AMP activity, which is the downstream signaling marker of GLP-1 receptor activation. And so theoretically, an antagonist shouldn't work if there isn't any ligand present. Okay, so part of the reason why we still don't know why this concept of GLP-1 physiology, intestinal GLP-1 physiology persists is because the model is really complicated. Typically what we do is we go to genetic models, we create knockout mice, and we can study our system of interest. But GLP-1 is more complicated because it's encoded from the pre-proglucagon gene, which actually makes many different peptides, as you can see here on the upper schematic. And the way that it does this is due to post-translational processing. So for example, pre-proglucagon is expressed in the gut, but it's also expressed in the central nervous system. And because of this prohormone convertase 1,3 is dominantly expressed there, you get production of GLP-1 as well as GLP-2. And at least in terms of intestinal GLP-1, we think that has a very potent effect on restraining postprandial glucose homeostasis. But interestingly, when you look at the pancreas, where pre-proglucagon is also expressed, another prohormone convertase is more dominantly expressed, and that leads to the production of glucagon, which interestingly has an opposing effect on glucose. So glucagon's role is to increase blood glucose levels, and it's often thought to work under times of fasting and stress. And so the complication here is that we have this one particular gene that is encoding for two peptides that have diametrically opposed action on glucose homeostasis. And so it makes it a very complicated system to study. But we went ahead and addressed it by generating a pre-proglucagon null animal, and the way that, or essentially a pre-proglucagon knockout animal. So the way that we did this is that we inserted a transcriptional blocking cassette, indicated here by the stop box, inside exons 2 and 3 of the pre-proglucagon gene. And so this makes an animal that doesn't make pre-proglucagon anywhere in the body. Now, one of the genetic tools that we use is that we inserted these LOXP sites around the transcriptional blocking cassette. These LOXP sites, represented by these green triangles, are recognized by an enzyme called Cre recombinase. So what we can do now is we can take this pre-proglucagon null animal and we can breed it to various promoter mice that express Cre recombinase in a tissue-specific fashion. So in the case of the data I'm going to show you next, what we did is used an intestinal promoter. So villain-1 Cre mouse expresses villain-1 throughout the intestine and even in the colon. And so what will happen is that the Cre recombinase will recognize these LOXP sites and remove this transcriptional blocking cassette. But because we're using an intestinal promoter, it will only do this in the intestine. And so what we'll have is reactivation of the pre-proglucagon gene specifically within the intestine, but it still remains knocked out in the brain and the pancreas. And so the graph that you see here on the bottom panel is actually demonstrating pre-proglucagon expression relative to the control animals in the blue bars. The pre-proglucagon null animals would be in the yellow bars had they any detectable pre-proglucagon, but you can see across all 4 sections of the intestine, within the pancreas, and within the central nervous system, there's no detectable pre-proglucagon expression. So we have effectively generated a pre-proglucagon knockout animal. But you can see in the intestinally reactivated animals, we now have a restoration of glucagon shown in the white bars in all 4 sections of the intestine, but not in the pancreas or the hindbrain. And so one of the things that we have, I will be honest with you, these genetic models, they always seem so easy when you read about it in the Nature and Science publications, but they're actually quite difficult to validate. And so we got burned early on some of the models that we used, and so one of the really important things that we do is validate both the plasma and protein and tissue protein levels of GLP-1. And so for that we need some nice assays in order to do that. So here is demonstrating that the intestinal reactivated animals have a restored postprandial total and active GLP-1 response to a meal. And so, again, in the pre-proglucagon null animal essentially has undetectable GLP-1 levels. But we also looked at the tissue protein levels to make sure that there wasn't any protein that was still being translated or still being produced but trapped in the tissue. And again, you see in the pre-proglucagon animal, there's no detectable GLP-1 levels in the duodenum, the ileum, or the pancreas, but we get a nice reactivation in the intestine, represented by the duodenum and the ileum, but not in the pancreas of those intestinal reactivated animals. We also looked at glucagon levels in the pancreas, and so we don't see any genetic compensation where the lack of pre-proglucagon or the turning on the pre-proglucagon in the intestine has any effect on the genetics of the pancreas. So intestinal glucagon was undetectable, so we don't see a compensation in that regard either. So normal glucagon levels in the Vilcree animals, the control animals, but undetectable in the null and intestinal reactivated animals. Okay, so I'm not really going to show you any of this data. It's actually currently being in revision, so hopefully look for it soon. But really, what I wanted to just highlight was the importance of using these types of assays that ALPCO has for validating these genetic models. And so questions that my lab has moving forward is, what is the role of intestinal GLP-1 in regulating glucose homeostasis? Is it sufficient but also necessary for regulation of glucose homeostasis? And the other thing we can ask about is, what is the role of brain and pancreatic pre-proglucagon in glucose homeostasis as well? Importantly, we need to understand more about those pre-proglucagon neurons in the central nervous system, and what is their role in regulating both satiety, body weight, and also glucose homeostasis. In addition, there is evidence that GLP-1 is made in the pancreas, and so we want to understand more about the functional implications of that source of GLP-1. And so, again, questions that we have moving forward is intestinal GLP-1 necessary for blood glucose? And if it isn't, then what is the function of GLP-1 secreted from that source? And does the pancreas produce GLP-1, and what is the influence of this source of GLP-1 on physiology, or is it simply just pathophysiology? And lastly, again, we really need to understand what is the role of GLP-1 produced in the central nervous system. And so, again, coming back to this idea of using these genetic models to try to understand the implications of GLP-1 therapeutics or bariatric surgery, that's actually what I'm going to focus on next, is the impact of GLP-1 on the success of bariatric surgery. And so it was noticed many years ago that with Roux-en-Y gastric bypass, a specific type of bariatric surgery where you have intestinal rearrangement, but also stomach size reduction, so nutrients are coming in and bypassing 95% of the stomach and the upper GI tract. But it was noticed that with this surgery there is a huge increase in GLP-1 postprandially, and so it was often implicated as a mechanism for the success of surgery. And so two questions that I'm going to use some data and to try to address today is, what is it that regulates this increase in GLP-1 bariatric surgery? And importantly, is this increase in GLP-1 necessary for the success of surgery, or is it simply just a marker that physiological changes have occurred? Okay, so there are many different flavors of bariatric surgery. I'm going to focus on the next few slides on two in particular, and that would be Roux-en-Y gastric bypass, which I've already described to you, but also vertical sleeve gastrectomy. In vertical sleeve gastrectomy, 80% of the stomach along the greater curvature is removed, so it's completely removed from the peritoneal cavity, whereas in gastric bypass, the stomach and the upper GI tract remains in the peritoneal cavity. Remarkably, even though the anatomy of these two surgeries are drastically different, so the intestine is not rearranged with VSG at all, they have very similar efficacy. And so with both of these surgeries, and these are optimistic results, but it's by far more effective than, probably 3 times more effective than any behavioral or behavioral plus pharmaceutical intervention. But you get about a 30 to 25%, 25 to 30% weight loss over a 5-year period, so meaning that that weight loss is sustained over time. And you also see diabetes resolution in these patients. So again, these are optimistic numbers at 81%. A more conservative estimate would be closer to 38 to 41%. But still, no other treatment can claim that kind of resolution from type 2 diabetes. And importantly, these two surgeries are much more effective than adjustable gastric banding, where you're just having a band placed around the upper stomach. And in that surgery, you typically only see about a 15% weight loss, and again, this is an optimistic number of 61% type 2 diabetes resolution. So for those reasons, we have really tried to compare and contrast these two surgeries so we could figure out whether are they really working similarly or are these two different mechanisms that lead to similar outcomes. So this is our rat model of bariatric surgery. Typically what we do is we put the animals on a high-fat diet for 8 to 12 weeks prior to surgery, and then we are able in our animal models to include a sham surgery group, not something we can get patients to volunteer for. And then, so what typically happens with these guys is they'll show a very early weight loss, and then rats continue to grow throughout their lives, and so they'll go on their normal growth trajectory. Whereas VSG and Roux-en-Y will follow a parallel trajectory, they never actually intersect. So they maintain this lower body weight. So this is, again, different than what we see in humans, but it's still, I think, an appropriate model to try to get at mechanisms of action. So one of the things, again, that we're very interested in is the impact of these surgeries on glucose homeostasis. And one of the interesting things that we noticed when we first started with these models is that when you give the animals a glucose gavage, so you administer glucose orally, you see that the peak glucose is actually really high with Roux-en-Y gastric bypass, and then it returns rapidly towards baseline. Whereas the sham animals, who, I forgot to mention this, but all of the animals are still on a high-fat diet after surgery, so we don't change their diet after surgery. And so what we're doing is really isolating the impact of the surgery itself, whereas with patients, you hope that they actually do change their diet after the surgery as well. So when we first saw these data, we thought it was surprising, but then when we thought about it a little more, nutrients with Roux-en-Y gastric bypass, they're pretty much being dumped directly into the intestine, so it kind of makes sense that you would have a very high peak glucose level. But the interesting thing was that when we did a head-to-head comparison with vertical sleeve gastrectomy, we saw this same trajectory of glucose. And it was surprising because the pylorus is still intact, and so we thought that the pylorus sphincter would still gate nutrient entry into the intestine. So we decided to go ahead and measure the gastric emptying rate, and so this is an image of the GI tract using barium. So we gavaged the animals with barium plus Ensure so that there was actual nutrient load mixed in with the barium. And this was an image taken 5 minutes after the gavage, and I'll explain why we took such a rapid image in just a second. So you can see this white here, so the blue outline is the stomach, and the white reflects that the barium is still well within the stomach. However, as you would predict, with Roux-en-Y gastric bypass, the nutrients are well within the intestine. However, in vertical sleeve gastrectomy, we see exactly the same thing. The barium is well within the intestine, and again, this is within 5 minutes. And so when we quantified this, we actually saw that 100% of the gastric contents were emptied within 5 minutes of both Roux-en-Y and VSG. And so we wanted to understand why this was, or understand whether this rapid emptying actually could directly influence the GLP-1 responses. So what we did next was we took the animals and we, so this is just sham animals, sham surgery animals compared to vertical sleeve gastrectomy. And this is active GLP-1 measured at baseline or 15 minutes after the gavage. So in the sham animals, we don't have much of an increase here, and that is because the dose of glucose is actually really low. And so we don't have much of a stimulus in the sham surgery animals, and yet we still see the nice significant increase with vertical sleeve gastrectomy. And again, this is when all the animals are getting an oral gavage of glucose. So what we wanted to do next is take these same animals and control the rate of gastric emptying. And the way that we did this was instead of gavaging the glucose, we completely bypassed the stomach and bypassed gastric emptying and infused the glucose at the same exact rate in both the sham and the VSG animals. So now glucose is entering the system at the same exact rate, and the question is, are we then preventing this increase in GLP-1? Because if we are, then that means that that rapid gastric emptying rate is what's driving the increase in GLP-1. However, we saw exactly the same thing with the duodenal infusion compared to the gavage. And again, this is a very small dose of glucose. We use the same dose of glucose in the duodenal infusion as we did in the gavage. And so even when we control the rate of glucose entry into the intestine, we still get this nice increase in GLP-1. So the other thing that we did is we've done a dose response curve of glucose. So these are, again, these are very low levels. So in the sham animals, you don't see much of an increase in GLP-1 because the caloric load is pretty small. However, with vertical sleeve gastrectomy, already at the lowest dose of glucose, you have a maximal GLP-1 excursion, right? Whereas with Roux-en-Y gastric bypass, you get this steady increase over time. And so, and then this is just demonstrating that when we finally got access to the ABCO kit, we were able to measure active GLP-1. The previous slide shows you total GLP-1. And so when we're able to measure active GLP-1, we also see the greater than 10-fold increase in GLP-1 that you would predict from Roux-en-Y gastric bypass and vertical sleeve gastrectomy. Okay, so what we know is that gastric emptying rate is being increased by both Roux-en-Y gastric bypass and vertical sleeve gastrectomy, and they both lead to an increase in GLP-1. But what we think is going on with vertical sleeve gastrectomy is that you actually get an enhanced nutrient sensitivity within the intestine, and that is actually driving the increase in GLP-1. We hypothesize that this is completely downstream of the increased gastric emptying rate. So if we could somehow slow that gastric emptying rate, we would no longer see that increase in GLP-1. So in other words, we believe it's a long-term adaptation to the continual increased gastric emptying rate that occurs with every meal. But you'll have to stay tuned as we try to probe a little deeper into the mechanisms that might be causing that. So the second question that I wanted to address is whether GLP-1 was an underlying mechanism for bariatric surgery. And so we have a couple of tools at our disposal that we can address this question. And so the first is pharmacological. So we can use we can administer a GLP-1 receptor antagonist, and we know that this, from some separate data, we know that this antagonist is very specific and very potent. And so this is the increase in insulin with vertical sleeve gastrectomy and Roux-en-Y gastric bypass compared to sham animals, and also compared to pair-fed animals. Pair-fed animals were actually given the same amount of food ingested on a daily basis by the Roux-en-Y gastric bypass animals. So they actually weigh pretty similar to these bariatric surgery animals, but you can see that their insulin excursion, which by the way looks very similar to the glucose excursion with a rapid peak and return towards baseline with bariatric surgery, in the pair-fed animals you don't get that same adaptive response. So it really shows that it's something about the surgery and not the weight loss causing this effect. So what we did is we gave a GLP-1 receptor antagonist and asked whether this could blunt this insulin excursion. Enough, we do see that Exenatine can block that insulin excursion. So these data suggest that GLP-1 is necessary to cause this increase, at least acutely, in insulin levels. So to really find out whether and these are acute studies, it's difficult to administer and also difficult to know what dose of Exenatine you should administer chronically. So we turn to genetic ablation of the GLP-1 receptor. So this is GLP-1 receptor knockout mouse. And we did vertical sleeve gastrectomy in these guys. And so here shown in the open and closed symbols are wild-type animals given sham and vertical sleeve gastrectomy respectively. You can see that the glucose curves are a little different in mice, but we still have that high peak and then rapid returns towards baseline in the wild-type animals. Now, animals that don't make the GLP-1 receptor, that don't have the GLP-1 receptor have impaired glucose tolerance, and we're demonstrating that here in the sham animals have a greater impairment or greater excursion of glucose compared to the wild-type sham animals. And so the question is vertical sleeve gastrectomy able to improve glucose homeostasis in these GLP-1 receptor knockout animals? And not only did it improve it, it actually normalized it. And so at least in terms of chronic ablation of the GLP-1 receptor, that receptor does not seem to be necessary for the ability of vertical sleeve gastrectomy to improve glucose tolerance. You can see that the basal glucose levels are lower as well with vertical sleeve gastrectomy, even in the knockout animals. Now, the data does seem to suggest a slight blunting of the insulin excursion, but the important point is that overall glucose homeostasis is still improved even when you don't have the GLP-1 receptor around. I don't have time to show you, but these guys also lose plenty of weight. They lose the same amount of weight, and it's specific to body fat as their wild-type counterparts. And so when you do the pharmacological ablation, you certainly can blunt that insulin response to an acute glucose stimulation after bariatric surgery. But the genetic ablation, those animals still seem to have overall improved glucose tolerance. And so coming back to the two questions that I started this section is, what regulates the increase in GLP-1 bariatric surgery? Again, we hypothesized that changes in nutrient sensitivity drive that increase in GLP-1. However, it seems that that increase in GLP-1 is just a marker for the physiological changes that occur with surgery and are not necessary for the overall improvement, for the weight loss for sure, but also the overall improvement in glucose homeostasis. And so with that, I will close. I should acknowledge all the people that actually do the work in the lab. So Adam Chambers, who's now at Novo Nordisk, did some of the bariatric surgery work, my technical support when I was at University of Cincinnati, and now my laboratory at University of Michigan. Of course, all my collaborators in generation of the genetic and bariatric surgery models. Thank you.
Thank you so much, Darlene. Thank you for sharing with us some of the wonderful data that you just presented. Very exciting stuff. I know I have a couple questions, but just to keep the momentum going, I think I'm going to hold off on them. And I'm going to switch gears and talk about the STELLUX chemiluminescence GLP-1 assay that you introduced in your presentation. All right, so just talking about or thinking about some of the challenges that come with measuring GLP-1. What are some concerns from a scientific standpoint? And Darleen actually hit on some of these in her presentation as well. One of the ones is just sample size, right? A lot of the folks that do study rodent models want to be able to use assays that don't require 50 or 100 microliters of sample size because it's just too much. The second is sensitivity. Since GLP-1 gets degraded so quickly, by the time it reaches circulation, levels are extremely low. And so you want to be able to have assays that can accurately and precisely quantify fasted GLP-1 levels. And then, of course, accuracy and precision. There are quite a few assays on the market that may not necessarily correlate with one another because they may not be accurate. They may not have enough spike and recovery or dilution linearity data demonstrated in their assay, which is why, when we were developing this assay, we wanted to make sure that we had squared away a lot of these issues that were currently being observed by a lot of the research folks that were measuring GLP-1. So what is STELLUX? For folks that don't really know much about it, it's a chemiluminescent ELISA platform that was introduced in 2013 here at ALPCO, and the goal is to really be able to detect biomarkers in the diabetes and obesity space, to be able to offer assays that can detect biomarkers with that have a broad dynamic range, that have superior sensitivity. We do provide extensive validation packages that demonstrate dilution linearity, as well as precision data and accuracy. And a lot of the assays on the STELLUX platform require small sample volumes, so that's very advantageous for folks that are, again, studying rodent models. So there's really nothing different from a protocol standpoint. It's like any other ELISA, but instead of, say, a TMB substrate, it uses a glow, a luminol or glow substrate, and at the end you develop or read the assay using a chemiluminescent plate reader. So having said that, let's dive a little more into the actual assay and some of its characteristics. It is a low volume assay. It's highly sensitive, very specific, and we've done enough characterization to show that it's consistently accurate. So low sample volume. What do I mean by that? This is the only assay on the market with a 10 microliter sample size, which is, once again, perfect for folks that are doing a lot of the rodent studies. There are a couple competitors, as you can see on the screen, anywhere from 25 to 100 microliters of sample size. But once again, the ALPCO chemiluminescence kit only uses 10 microliter sample size. It is highly sensitive, so we've tested extensive, samples, both mice and rats, both fasted and fed levels, and you can see that sensitivity of this assay is less than 1 picomolar, which is great if you want to measure fasted active GLP-1 levels. And then it is also highly specific. So this is a kit that specifically measures GLP-1, the 7-36 amidated form, and no other species are picked up in the ALPCO assay. Whereas if you compare that to some of the other assays on the market, you will note that, it does cross-react with one or two or a couple other GLP-1 species. So this is important because the 7 to 36 amidated form is really the biologically active circulating form. What about accuracy? We've done, like I mentioned earlier, we've done extensive both dilutional linearity as well as spike and recovery studies to make sure that the assay is consistently and accurately detecting only GLP-1 levels and that there are no other interfering matrices. Or any other interfering reagents in the matrix that are being picked up and that may interfere with the quantification of GLP-1 7 to 36. And so just to summarize, this assay is already on the shelf. The catalog number is 80-GLP-1, GLP-1A-CHO1. It does cross-react with human, mouse, and rat, so you can detect human, mouse, rat, EDTA plasma, as well as rodent serum. Once again, it only uses 10 microliters of sample. The range is quite wide, between 0.76 to just over 1,000 picomolar. Sensitivity is less than 1 picomolar, and incubation time or total assay run time is about 2.5 or just under 2.5 hours. So it's great compared to current assays in the market that may require overnight incubation and hence have a longer turnaround time. We have done a lot of collaborative work with, obviously we've done some work with Darleen at the University of Michigan, but we've been heavily involved with a bunch of different institutions, including the Holtz Lab at the University of Copenhagen, UBC, University of Alberta, as well as the Oregon Health and Science University. And with that said, I'm going to open up the session for any questions that folks may have. I've also included some contact information in case you need to reach out to me for more information on GLP-1 or any of our other assays. So with that said, please feel free. The question session is open. Darleen, you mentioned the role of intestinal GLP-1 and how it's necessary for maintaining glucose, blood glucose levels. What are your thoughts on quantification of total versus active GLP?
So of course I think it is more important to have active GLP-1 levels over total. Just because of the rapid inactivation. And I think actually some of the, I didn't show this data because we're getting ready to publish it, like I said, but our data, our genetic models actually suggest that intestinal GLP-1 is not necessary for glucose homeostasis, that it is actually pancreatic GLP-1. And so it really questions this endocrine model of GLP-1 function. And I think part of, and as I've been presenting that data and some of the reviewers' comments that I got back were really reflective of, I think, because the fact that people often measure total, they are under the assumption that GLP-1 is in the system longer than it actually is. And so, so I think actually it may be the fact that it's what we can measure right up until now, and so it's the data that we have. But I think the ability to measure active more accurately is going to be pretty important moving forward.
Thank you. Since we're on the topic of total versus active, when do you think it would be important to measure total GLP-1? Or even
So,
both as simultaneously?
yeah, no, I actually think perhaps both, right? Because if there is a situation, a pharmaceutical situation, so for example, the DPP-4 inhibitors, if you want to look at the ratio of the two, that might be informative for looking at action. If there's any pharmaceutical that is somehow impairing hepatic clearance of GLP-1, then you might want to know total under that. You might want to know the ratio under that circumstance. I think also understanding the timing. So if you've given a meal and looking at the change in the ratio over time might be informative as well.
Right, and you think that that would really depend on the species, right? Because it's
It could, yeah, definitely.
differently in different species.
Right, right.
Okay. That makes sense. Thank you so much. I would like to thank you all for participating. Thank you so much, Darleen, for taking the time to share some of the stuff that you've been generating in the lab. Once again, should you need additional information on any of the assays or even specifically the GLP-1 assay, please feel free to reach out to me. My contact information is on the screen. And with that said, I am going to end this webinar. Thanks again, and have a great rest of the day.